Hello.
Am I audible?
Yes.
Okay.
All right. How are you?
Yeah, I'm good. Yes.
All right.
I've been talking to Mehul also regarding my proposal,
Okay.
and since this is the first time that attention is being used in the DevoGraph library, if I'm gonna use it. So I also wrote a small blog about it. I think I also shared it to you over the Slack channel.
Yeah. It'd be good.
So yeah, it's in my write-up. Like I wrote, it consists of like 5 papers which I went through, and like how each of them use attention in the field of developmental biology. So I'll also include that in the proposal as well, like a small literature review.
Okay. Did you have the blog post? Did you want to share
I'll
it?
share it in this chat.
Okay. So yeah, this is attention in developmental biology, and with a question mark. All right. Spatio-temporal AI and graph learning. Some papers I went through. Yeah. Here's the reality: most biological systems are not static. Brains evolve, cells communicate, organisms adapt. Yet many traditional AI models treat the world like a frozen snapshot. That's true. These 5 papers challenge that assumption, and in doing so, they're reshaping the entire landscape of spatio-temporal AI and graph learning. Okay, so you're going to break down 5 papers. This is good. I like this. The first is SGEL, EST-GEL. That is understanding how brain networks evolve. So the SGEL stands for explainable spatiotemporal graph evolution learning, and with applications to dynamic brain network analysis during development. And so the big picture is that imagine your brain is a living, breathing network of connections. It's not the same today as it was when you were 10, and it won't be the same at 60. And SGEL captures how the brain network structurally and functionally reorganize as we grow. So this is a good one. So this is the SGEL method.
Yep.
Okay, and then this is so it works by using edge attention modules, dynamic gating mechanisms, and explainability built in. And so the key insight, if we use MRI data, SGEL reveals that as we age, our brain networks transition from highly dispersed to concentrated efficient structures. Yeah, this is really interesting, and I know you presented on this particular method.
Yeah.
And this is very much like a complex network where you have things like preferential attachment and other types of mechanisms to go from a random network to a structured network. So oftentimes in complex networks, they'll analyze a network in terms of how random it is or how ordered it is, and they'll use some framework like they'll say something is scale free or something is small world, and they'll use that as sort of the ideal, and then they look at sort of the sort of where it is in between those ideals. So a lot of networks are not random. The ones that you find in nature, they're not random, but they're also not idealized small world networks, idealized scale free networks. They're somewhere in between. So that's an interesting approach because it does relate more to the kinds of networks we see in the real world. So yeah, from a developmental biology perspective, this is huge. For organisms like C. elegans, we have the complete connectome mapped, but we don't fully understand how it changes over development. So the second method is ASA. This is the beauty of simplicity in graph AI. This is the paper here is end-to-end attention-based approach to learning on graphs. So this is the edge set attention you presented on. And then graph neural networks are notorious for their complexity, positional encodings, message passing schemes, pre-processing pipelines. That's a lot. Yeah, I think the one important point here is that we'll use a positional encoding, and then that sort of makes the network static in a lot of ways, because you kind of give like either like a specific data set of positional encodings, or you hard code positional encodings, and it's kind of like there's no spatial variation there necessarily. And message passing schemes are, of course, important, but they do kind of take their inspiration from communication networks. You know, like if you had something like the internet, where you had some sort of communication network where you're sending messages across it. And in biology, you do have signaling, you do have things like that, but using message passing schemes there seems kind of artificial in a lot of ways. So ESA throws most of that away and proves that sometimes simplicity wins. So edge set attention works by treating a graph as a set of edges or connections between nodes. It uses pure attention mechanisms, and then alternates between masked attention, where it only looks at directly connected edges, and self attention, where it sees the bigger picture, and that's it. And then despite being designed for simplicity, ESA outperforms heavily fine-tuned complex graph transformers across dozens of benchmarks. And of course, we can use this for a number of things. And it says it proves that architectural elegance often defeats brute force complexity. So that's, again, give it. Sometimes you just need to focus on the right things instead of including every detail. Number 3 is self-referential GNNs. This is networks that evolve themselves, or graph neural networks that evolve themselves.
One more paper that I found out about hypergraphs. So I thought about including this also.
Okay. Oh, is that in the post, or is this a separate one?
No, it's in the post itself.
Oh, okay. Yeah. So this is on hyper networks, that evolve themselves. And then what if your AI model could literally mutate itself? So we get, this is like kind of almost like including a genetic algorithm type approach into graph neural networks and hyper networks. So and so this is self-referential graph hypernetworks, or GHN. This embeds evolution directly into its own code. The network looks at its own structure, generates mutated versions of itself, tests them in the environment, keeps the winners, discards the losers. The network becomes the optimizer. So this is where they show this computational graph of self-referential GHNs. I think we talked about this paper too, because I remember this graph. And this is where you have the self-referential evolution. Okay, so this is the key point here, is the network exhibits emergent control over its own evolvability. So evolvability is the ability to evolve or to reach different solutions given its like artificial genome. So if you have like an artificial genome, sometimes you can get to certain states and be a stable or a viable solution, and sometimes you can't. So evolvability in a system is important. This is a topic that people have talked about a lot with organisms being able to evolve themselves, into specific niches, or being able to adapt to different environmental conditions. So the 3 things focused on here is whether or not the environment's stable, which reduces the mutation rate. When the environment changes, it cranks up the mutation rate. And when there are novel challenges, the network figures out it needs to change strategies. I assume this is a very specific to this method. This is the first step towards truly autonomous open-ended learning agents. And so this is I can't remember who the authors were on this paper, but I remember this paper quite well. Number 4 is Cell STM, the ultimate cell tracker. So this paper is Spatio-temporal feature-based deep learning, or deep neural networks for cell lineage analysis and microscopy images. And so this is where it kind of focuses on cell tracking, and maybe some questions that someone might have in observing a cell tracking data set. So where does a cell move? Where did it divide and where did it die? And so you want to have timestamps for those. You see things in the cell tracking, but you don't know how to interpret it. So for example, if a cell disappears, is it dead or did it divide or did you just lose track of it? Which one is it? So cell STM automates this nightmare. So cells aren't static objects. They change shape, move, and behave unpredictably. Standard computer vision isn't enough. Cell STM uses bidirectional convolution LSTMs or conval STMs. This scans video frames from both forward and backwards in time. We have multitask learning, which predicts the locations, divisions, and deaths of cells simultaneously. And then visual plus temporal features, learning both what cells look like and how they move. And so this is where we get an AI that understands cell behavior in a holistic way. So again, this is where we have an initial position, we have a spatial representation, we have an input sequence of positions, we have this encoding, which converges upon a prediction from the spatial representation and the encoding from the input sequence. We concatenate them, and then we have this attention branch, which focuses on this solution using a conval STM. So that's what we're doing here, and they call it the temporal cell cracking, which is like just the term that they use. Here's what makes it special. It only requires a weakly supervised coarse set of labels. No need to perfectly trace every cell outline. And for developmental biology, we could use cell STM to track cell divisions, understand cell migration patterns, study developmental abnormalities, and other things as well. The number 5 is where we have cell nest, decoding cellular gossip. So this is where we're looking at cell-cell relay networks, which are signaling centric models. And so this is where maybe we can replace message passing or some other technique that's not necessarily biologically specific with something more biologically specific. So this is their approach. Cell nest. Cells communicate through a language of proteins or ligands that bind to receptors. So we have all sorts of signaling that goes, that's intracellular, that one cell sends another cell, and they have to receive that message through a binding site, and then that's, that ensures communication between cells. So we get these networks of communication. And so CellNest can decode these relay networks. So using graph attention networks, and so, I guess it's GAT. I guess they don't want to confuse it with GANs. But yeah.
Yeah, that's okay.
Yeah. So using GATs trained in an unsupervised manner, CellNest analyze spatial transcriptomic data, identify which cell communicates with which, detects relay patterns. So if you have something transitive like A is to B, B is to C, and then maybe C is to A, or you just have this relay pattern from A to B to C, you can detect that. And then it outputs high confidence communication maps from that. So this is an example here where you have this image here, and they're extracting this cell relay network, which just maps out the interactions between cells, the direction of the signal, and it's all predicted from, I guess, temporal data and the presence of so I may be like they're using fluorescence microscopy here as well to figure out the distribution of these ligands. So you're getting this you can get images over time that show how this moves around, and you can figure out where this which cells are connected to which in that sense. So they have stain. They're using a stain here where they have an image that's a stain, and then they have the time series of stains, and they can see how that signal diffuses or moves across the anatomical structure, and then they're able to get sort of some sort of directionality. So it's kind of like what we're talking about with Lagrangian coherent structures, where we have these, this diffusion of particles, and our job is to track the divergence of particles and how they form these ridges or other structures. You just do the same thing here, where you have the dark spots on this microscopy image, and they're essentially concentrations of molecules that diffuse from central points, and their direction of diffusion tells you which cells are communicating with which cells. Something like that. So this just goes over cell nest, this figure, and then the real world impact of cell nest is that you can look at things like T cell homing signals in lymph nodes and aggressive communication networks in tumors, where cells are signaling each other quite a bit. And that's really the basis for sort of metastasis. And then, of course, with T cells, you need different signals that coordinate their migration and tell them where to go. And so this is interesting because we can model not just directional signaling, but like information cascades across tissues. Okay, so what connects all 5 papers? One thing is that modern AI for biology isn't about finding static patterns. It's about understanding dynamic interconnected systems. Yeah, I think that's true. And the respective domains are all treated as living, changing systems, not frozen snapshots. And then Barshan put in some things about his GSOC work and what his proposal is, and then some final thoughts. Yeah, I like that. And is that something you do a lot of blogging on these kind of papers, or?
No, it's just that I have a blog where I write experiences about papers or about my projects. So
Okay.
This could use a literature review, so I thought about putting it in my blog as well.
Yeah, I think it's great. Yeah, thank you. Thank you for sharing that.
Thank you.
Okay. So did you want to present on anything, or?
I just had a literature review. I'm working on the proposal as well. And other than that, I looked through the data set, the EPIC data set. So understanding all the parameters that I have available. So I understood 2 of them, like blob and G weight. So G weight is the one that has the weight of the genes, and that is used for clustering purposes by the simulation softwares. And other than that, blob is the some intensity value, which can be used as a parameter in my tensor, which I happen to work on. So that's what I looked through the weekend.
That's good. Thank you.
Thank you.
All right. So it's Barshan. Thank you, Barshan. We have Susan, we have Ashu, Utkarsh, and Morgan. So I don't know. Susan wanted to give an update.
Sure. I was playing with Gemini.
Okay.
This weekend.
Yeah. I saw your email.
Yeah, and it took the diagram from my thesis and gave me the computer code for it, the connectivity matrix, and wrote me the code in Python and MATLAB. And then it evolved and showed me how to work with cell sheets, how to make more parts to my diagram. And then it changed my trijunction from a T4 to a T6 tensegrity model, an isohedron of some sort. And he said that was superior because it didn't get stuck in certain places. And you'd have to check that out. But I could go back in there and I could say that I wanted to do a 5-7 tiling and what's the difference between this for cell sheets. Like I could do a bunch of things. And it would give me the computer code and a bunch of leads into more research. So it was quite entertaining, especially the last part when it was giving me some a quantum build using the T6 structure because it would survive Planck's constant. And about then I was laughing.
Yeah.
It's like, sure. Anyway, yeah, so that was actually fun.
Yeah.
Because if you know enough about a topic, you can keep it from making the wrong
Right.
turn in the road, so to speak. You have
Yeah.
to kind of guide it, say, well, no, you don't want to make the structure, the T3, like the structure stronger. There's actually a middle structure, and then it says, oh, this is a good idea. It's a sandwich structure and it's stronger. And I'm going, well, yeah, that's the way it works. But yeah, I had to say no, make sure that you include the middle structure on this. And like I had to guide it a bit. Yeah. Right. You have to know enough about the subject in order to guide it. But it was making all kinds of computer code for me.
Yeah.
As it went.
That's the idea. Yeah. You have to check that though, because sometimes it doesn't work. We've been having an issue in our. Actually, we've been having an issue in our one of our Google Summer Code projects where people have been generating code, at least in part with AI, and it doesn't work. And so you really have to test it before you can implement it. I mean, I don't think in your case you're not pushing it to GitHub, so that's not.
No, I'm quite willing to try it out. Yeah. If it fails, I might look at why it fails, or I might just go back in there and say, this doesn't work.
Yeah, you have to do that a lot, actually. But it's
like, yeah.
Because like for scientific applications, if you're asking specific questions, you might ask it a question, it might give you some code, and then you look at it and you say, well, it's not really doing what I want. And you might ask very narrow, like you'll narrow your question. It won't be anything like a scientific question. It'll be like, why did you model it this way, or can you model it another way? And they're different. Like you can't get that unless you ask those very specific questions. So it's very much like if you were doing research and you were just writing code, and then you looked at it, you went back and looked at it, and you kind of thought, well, okay, this piece here, can I improve that? Or how does that relate to something in the literature? And are there better models for this? Or all sorts of things like that.
Please give me the sources that you're using to say this.
Yeah.
And that I found that always important, even in the other AI I was using. Anyways, yeah, please give me the sources.
Yeah,
that's always a good one, because then you can go back in, read the paper, and go, no, it kind of took a left turn when it was supposed to go right, and just
yeah.
But it gave me connectivity matrix for my project, and I'm very grateful. And I'll see if it works.
Yeah. Is it like like just generated it like from theoretical data?
Yeah. Okay. It generated it from an image I gave it.
Oh, okay.
A 3/4 view, and I had to correct it. I had to say, well, your measurements are wrong. This is a 3/4 view, engineering drawing. And so then it corrected it, and it had it correct. It had the right measurements.
Yeah.
From my diagram. Just a flat JPEG drawing.
Yeah.
It's just like, I'm amazed. And then I described the interconnects, and it got those right too. And it's like, all right.
Yeah.
Like, I'm really happy to have the connectivity matrix, never mind the rest of the cell sheet stuff it was doing. So yeah, it gave me several months work in 8 hours. So yeah, you have to check it.
Yeah. Oh, it's good. Yeah. Oh, Morgan had something to ask.
Yeah, there's a good interview with we've talked about this guy before. It's like something like Scott Williamson, but like Willison. I think it's Scott Wilson. He's like the original developer of like WordPress or something.
Yeah.
You know, and has been doing a lot of agentic coding. Anyway, he just gave a recent interview where he was talking about test-driven development, TDD. And if you know, sorry, I'm just getting my daughter ready for school, but it's interesting to look that up. You know, have the agents using a test-driven development can be really helpful for catching some of these issues. And also, I've implemented the Crunch Lab agentic SciML swarm, and it was interesting because so this is where the agents are working as a group, and they're like trying to, you know, have checkers as well as generators, right? And so yeah, it was interesting where they're going through, you know, so I can just leave things running on the computer and it's generating new experiments and you know, deciding the direction of that. So instead of doing grid search, it's like autonomously guiding the experiments. And it is, you know, like no like super results at this point, but it is good to see that it's definitely caught errors of the sort you're talking about, where it's like we're doing quantum experiments and it's like, oh, we seem to have a really good result, and then it's like, oh no, there's a, you know, the error checker is able to identify that it's a bug in terms of how this particular equation was you know, being set versus a different framework or something like that. Like yeah, so it is pretty remarkable, but you know, the guardrails are what he talks about, and he's more of a software engineer in terms of, you know, he's not interested in scientific, you know, scientific modeling, but he is talking about how the software engineering groups that are working this way are like the important part of their loop is identifying these kinds of issues and then figuring out, like, what do we need to put in place so that particular problem doesn't happen again, you know? So like the things that you were seeing, how would you change your prompts or your you know, guidance documents for agents such that they wouldn't make that same mistake again, you know?
Right.
Anyway, but he is talking about how it's still a very open area to try and figure out this. The best practices in this area.
Yeah, we've been trying to figure that out in the other project that we're doing with the large language models, where it's just like kind of people are putting out a lot of things. You know, you're proposing a lot of things, and then you want to integrate it into a pipeline, you know, some sort of workflow where you have where everything fits together. So like that's one of the problems with like a lot of software engineering teams in the first place is that, you know, usually you either have a top-down sort of workflow that you impose upon the project, or, you know, you kind of develop pieces very slowly and kind of work on feature by feature In this case, what you're doing is you're basically developing a huge, you're developing a huge platform very quickly, which is kind of the aim of Agile, right? Or something like that, where the promise is you can develop an app in like 4 weeks or something, and it's just magic. But in this case, you have a large volume of code you have to evaluate, you have to interrogate it because you're not, I mean, you have to do that anyways with developers, but like, in this case, we have to make sure that it's like, that, that it's of integrity. We have to do things like unit tests, but then we also have to fit it into a larger framework. Morgan's project that he's been working on, he's been working on this pipeline for neurosimulation, and he's got all these different parts. And so, I mean, I don't know, Morgan, how do you envision that? How would you envision a roadmap for that? Do you think that would be like, um, I mean, what's your roadmap? What's your thinking in terms of a roadmap for that?
Yeah, I mean, it is an issue, I mean, but it is an issue that like software engineering has dealt with before in terms of, large teams working where, one team is not necessarily going to do full code review of another team's work, right? So the question is, can you kind of specify what the interface is, And I mean, this is where it becomes more like, um, uh, kind of a distributed API development, where you've got different groups, but they're organized around some interface specification. It is, but it is harder because that's the kind of thing where there's kind of like less guidance and more just like best practices. Or it makes sense on paper, but it's really about organizing your project.
Yeah.
And holding the different groups to a bar, so it's like you can't, nobody can do that for you,
Right.
and so you've got to say what that is. And but I think, I think I'll try and drop a link later this morning of just in terms of that particular talk where he's trying to give you examples from teams that are working agentically where they've stopped doing code review, but that doesn't mean they've stopped trying to put guardrails on this.
Yeah.
So the question is, what does that look like? And yeah, so it's definitely trying to think of larger systems level about your tests, and that's maybe some of the test driven development work is helpful. Because I can't just use unit tests as a measure. I've been trying to come up with more code quality measures, as well as like trying to deep dive like what do some groups do with regard to that, like anyway. I'll definitely love talking about it when it's appropriate. But the DiPi developer, when we were talking about agentic coding, was talking about dealing. He's works with this massive open source project, and he gets a lot of code contributions, and he's both overwhelmed and unsure what to do. Because he's like, we're very much we've written all the pieces ourselves,
Right. Yeah.
or reviewed those, and how to move, when code generation becomes so easy, what are you going to use as your measures? and Terence Tao, as you know, the mathematician is very interested in working with coding agents. And he also sees like idea generation is now very cheap.
Yeah.
So the question now is like, how are you evaluating this at a similar scale? And
Well,
it's a lot of open questions.
yeah. Well, idea generation has always been cheap. It's just a matter of evaluating them is the problem. Anyone can have an idea, but
like Susan's saying, like something that would have taken you a good amount of time is now something that you can have the agent code up,
right?
And now you've got the bigger question of, okay, what did I actually want this to?
Yeah.
What did I want this to do? And how would I test that it's doing it?
Right.
Yeah, it just took off. It just took off in different directions. Like
Right.
I'd correct it and then it would take another tangent. And in the end, I just let it go.
Yeah.
And it ended up trying to contact the rest of the universe using my tensegrity at Planck's constant range. I'm going, really?
That's
it was hilarious.
yeah.
Okay, fine.
This is what they call the AI. This is the AI slop equivalent of SciML, where it's like it's reached, it's touched the sky. Yeah.
It's a whole science fiction book. I don't know.
Yeah. Well, it's
definitely
instant and
science fiction.
Yeah. It's instant and persistent scope creep, though. It's like, if you go off in these different directions, it's like, okay, I'll do this and this and this. Then all of a sudden you have this huge box of things that you are trying to follow up on. And then how do you write that up? How do you write that up in a way that like you can understand what you're actually doing? It's like you don't want to, there's a temptation to include everything in projects. We usually have ways to sort of prevent scope creep, in projects. And this isn't helpful. Yeah.
That's a good way of describing this. Scope creep.
Yeah, I think that's the.
It was fun. I was laughing hysterically halfway through.
Yeah, I mean,
yeah.
But even last night, like I was trying a different architecture versus normalizing flows, and it was iterating through. It's using an A100, and it's reporting this 28% gain, and I was like, yes, but that's a 28% gain, where you're still like 400% off what we were doing with a better architecture, right? It's got, it's still got this problem of kind of common sense that
Right.
you have to very, very carefully inject.
It's like the grounding problem, except a little bit more practical.
Well, I love the DARPA. You remember the DARPA team that was working on Common Sense, and their logo was an agent sitting in a tree sawing the branch that it was sitting on.
Yeah. Well, yeah. Thanks, Morgan, for that. That was good. Good discussion. It's like Utkarsh is here. Would you like to present or talk about your updates on things?
Hi everyone.
Hi.
I actually shared a repository in the chat here in Jitsi. I'd like to just explain what I was doing there, and also explain the overall MDG pipeline that I'm pushing forward in the proposal.
Okay.
If you could just check it out, and maybe either 8-cell simulation, it's at the results inside the repository.
Okay, so these are cells and they're in a position. They're dividing. Okay.
Yeah, right. Actually, this is a older version. I have a better one. This one was like the current one. These are each of them are cells as agents, and they are dividing. The graph here is like the connections, like which of them are physically connected, like touching each other. And below here is the contact area for the graph. Previously, I was using spherical units as cells, so the graph was completely broken. Like each cell was touching every other cell due to like the shape. So the correct solution would be using a vortex model, but it would take like around 1 or 2 weeks more, I would say. So for quick validation, I was just using a deformable cell. Each of these have like the 3 axes, 3 semi-axes, and they can elongate along those. And now, like previously, I had 28 out of 28 degenerate results I have mentioned in my report. Now it reduced to 7 out of 28, which is like pretty great jump without a vortex model. So that basically proves that vortex model was which is what it was missing previously. Yeah, this is it. Is there any question like regarding this?
Yeah, that looks interesting. So this is your repository, mechanistic. I saw this, and I was interested in what you're doing here. So yeah, is this a fork of DevoGraph, or is this kind of your own code here?
It is my own. Like I started this from like the ground up. I mean, let me.
Yeah, there it is.
Yeah, so this was the entire SINDyG pipeline which I was thinking about. Like I love building architectures from my own intuitions and basically like designing things on how I imagine them. So ABM is the first part of it. It has 3 reasons why it sits here. I mean, for now it seems like it is disconnected from overall like the connectome part of it. But like the 3 reasons are: one, that I wanted to see how like the cells would emerge, or the how overall physics would emerge if I just assign cells as agents and apply physical parameters to them. Like it was my own curiosity. The second reason was that I was trying to see, I mean, the results itself are really great. I was hoping that I would get something, but the animations I got are like pretty impressive for me. The calibrated parameters, there's no real way to validate them, since they are not like, there's no one value online that I can compare them against, but all of them, like most of them, lie in the correct range. The contact areas were like pretty decent. The AUC was 0.38 previously. Now with the vortex model, it would improve. And the last one, which is basically how it would connect to like DGNN or the HGNN part. Previously, I was using SINDy. SINDy uses the inputs from our ABM, the outputs from our ABM, the continuous physical parameters that it gives out, the positions X, Y, Z, and then it forms like the functions or the equations that would guide our NDP. But there's a problem with SINDy that it does not have any like knowledge about 2 different neurons that like work together and are connected basically. So I read a paper named SINDyG. It was like published in previous year, December 2025, and it was like targeting that exact same thing, where like 2 different nodes are connected and they are like together they are producing an equation. So it solves that part. Now I'll be implementing that over my ABM output. The problem with the actual like ABM here is there to validate whether SINDyG would work with that or not. Otherwise, I will have to like modify that I cannot use that directly on like the affiliate dataset. I will first try to validate using ABM. Then there's this, which is basically an algorithm that produces hyper edges based on the dynamics of data. Like both of these work directly on data, so it is important that I validate them. And ABM is like outputting the best one that I can actually compare against. So just with the Vivilate, I don't have anything to compare against. So I will use ABM to check whether they are working correctly. Then I'll modify them if they aren't, and then I get like the NDP table, and I also get an incidence matrix H, which I'll be using to build my DGNN or the HGNN. This was like my whole idea.
So the NDP table is a neural developmental program. So it's just producing different programs from the, so you have the ABM and you're kind of getting the best configurations for different cell positions or whatever. Putting it into SINDy, that's like the SINDy graph, which is the sort of the biophysical modeling of the graph. And then you're getting an NDP out of that. And then you're also doing the other part with the hyper edges, and you're producing the confidence matrix, and then you're putting that together.
Yes, that's basically it.
Yeah, well, that's great. Yeah, I was interested in that. I think that's a pretty good approach. Yeah, the NDP. You know, when we did this before, we just kind of created an NDP, like forked that repository that they had developed it in, and then we were working on building a graph from that. And you know, it's like it's computationally expensive to do that for very large data sets. So that's, I think that might be an advantage here to sort of cut down on that and get better representations and things like that. So yeah.
Actually, I read a comment on Neurostar. Somebody pointed out that the current HNN used like the GNN and had like set values. It wasn't like dynamic in nature. So yeah, I believe SINDy would solve that out in the current report.
Yeah, I think so. I'm not familiar with SINDy very much, but yeah, I mean, we'll see what happens in that, you know, trying it out like that. Yeah. Well, thank you, Utkarsh. It's very good work. Looks like Ashu is here. She would like to present, I guess.
Yes. Am I audible?
Yes.
So last week,
sorry.
Okay.
I just want to confirm whether my screen was shared. I was like presenting one image. I think I'll just share a image on Slack. Maybe you can check it out later.
Okay, that'd be good.
Sorry.
Yeah, that's fine. Okay, Ashu.
So I was saying that last week I spent most of my time in reading research papers. So there are some points that I want to discuss here, like about dynamic hypergraphs, vertex convolution, functional hypergraphs, and anastomosis and anastomosis loss. And there are two more topics like edge-dependent hypergraph neural network and spectral hypergraph analysis. So I have not made any specific PPT for that. Like I have made my notes, so I want to share them.
Okay.
Okay, so these are my notes that I was preparing for today's meeting. Like for dynamic hypergraphs, the insight is this: the hypergraph structure should be derived from the current node representation. Like we all know that the neural network grow by time. So the hypergraph should also evolve. Like so the dynamic hypergraph construction, this is the mathematical equation which will represent the current layer. Like the dynamic hypergraph should evolve layer by layer. So there are 2 points. Firstly, the local, which is GNN based. So for each node Vi with current embedding Hn finds its k nearest neighbor in embedding space. This is the equation, and in this the k nearest neighbor of a node is found, and the hyper H is made locally. And in other part, it is a global part in which it is k means based. Like we run k means clustering on all the current embeddings, the h1 till hn, to get k cluster center. So in this we get the hyper h for global points, and we will combine the local and global together to form a dynamic hyper h. It is important because currently there is a special hypergraph and the lineage hypergraph, and there are two more things that should be integrated in this: the functional hypergraph and the anastomosal hypergraph. Because there is another point, like in the it is vertex convolution layer is inside every layer of the hypergraph. Like in the dynamic hypergraph, it evolves layer by layer. So this vertex convolution is present in every layer. So if in the first layer, the hyper edges are made using the current data, and for the layer 2, the previous embeddings are also considered. And after building the dynamic hyper edges, the dynamic hypergraph neural network uses a learned transform matrix rather than the fixed aggregation. In this, the learned transform matrix from vertex features. This is a learned transform matrix and the 1D convolution layer, which vertex ordering and combinations are important. So there is a stack of the vertex feature. These are all the vertex feature, and it is a transform matrix. And in the step 2, we apply a transform and produces hyper edge features. So we apply the 1D convolution in the transform matrix, which have features this, and it learns the weight permuted combination of the vertex feature, because some weights are hyper edges matters more. Like suppose there are 3 types of cells, and they all connect differently from the other 3 cells. So it is important to know that which cell matters the most to define the feature of that hyper edge. And this is the functional hyper edge that should be integrated before the dynamic hypergraph. So the functional hyper edge connects embryonic cells that will collectively build the same neural circuit in the adult C. elegans neuron. It is not based on where cells are now or where they come from. It is based on where they are going. So it connects the cells that actually forms a adult C. elegans. So in this, the functional loss pulls co-current cells together in embedding space. After training for several epochs, cells destined for the same adult circuits are closer together in the 64-dimensional embedding space. So in this, the cells that are actually close in the adult C. elegans are put together in a 64-dimensional embedding space. And the attraction term for the functional loss explicitly minimize pairwise embeddings. So in this, the functional loss we found it by the attraction loss plus the lambda r, which is the relative weight of repulsion, and multiplied by the loss of repulsion. So we find the functional loss by this formula, and the L attraction means the attraction terms, and this means the repulsion terms. So we find the repulsion term and the attraction terms, and by that we will find the loss function. And the dynamic hypergraph neural network reads the spaceship. When dynamic hypergraph neural network rebuilds hyperedges at layer 2 of the forward pass, it runs GNN on whatever embeddings currently exist. After the functional loss has reshaped those embeddings, the dynamic hypergraph neural network GNN search naturally finds the co-circuit cells that are close together. So after the functional hyperedges are known, like the edges that are actually closer in the adult C. elegans are known, it is to find the dynamic hypergraph neural network, because it will find the embeddings layer by layer. And another part is the anastomosis and anastomosis loss. Like anastomosis in the context of C. elegans development means the cell crossing from one spatial cluster to another between consecutive time points. And it will be done by finding the anastomosis loss in the training objective that teaches the neural network to predict which cell are about to switch cluster. Initially, when the in the embryosis stage, the cells divide. Some cells are in different clusters and they switch their clusters while growing. So this finds that which cell is switching its cluster by time. So it is we will find out by defining the membership probability. Like in this, we will find the which cell and hyper edge are probably having the most membership probability, like which cell and hyper edge will be together for more time. So it is found by this equation, and the maximum membership probability of cell I across all hyper edges it belongs will be find out by this equation. And the pi max, if the pi max is close to 1, the cell is firmly embedded. Like means that if this term is close to 1, the cell is connected to a similar cluster. Like it won't change its cluster by time. It is very tightly embedded to its current cluster. We will find the functional loss by the binary cross entropy between the predicted memberships. Yes, sir.
Oh, that's good. Thank you.
And sir, there are two more things that I have not mentioned here, but I was reading, but since I don't have a very much knowledge about them, so I didn't mention here. First is edge-dependent hypergraph neural network that will tell that information flow from one node to another through a hypernedge. And the second is spectral hypergraph analysis. So in the edge-dependent hypergraph neural network, it is like if a hyperedge has 3 or 4 nodes and like neuroblast, muscle, and similar, and what it should, like the cell is connected to a spatial hypergraph, to lineage hypergraph, and functional hypergraph and anastomosis hypergraph. So the cell should not pass the same information to all the 4 edges. Like it should pass the different information, because it is not like in biology. It is like this, that same information is not passed to all the cells. Like what is important for special lineage cell, that information should be mentioned, and what information is important for the lineage cell should be passed only that. And in spectral hypergraph analysis, it I have not very much knowledge about this, but it is like it will be integrated in the dynamic hypergraph, like in layers, to find the loss function, I guess, but I'm not sure about it. I will probably do more research on this specific topic.
Okay, yeah, thank you. That was good. A lot of good stuff there. So we had, yeah, I think you stated a lot of those things very clearly, and yeah. So yeah, I think that's good. I'm just looking over my notes because you had a lot of stuff there. Yeah, I liked how you kind of laid out the hypergraph and kind of what you have the global part and the local part, and then you're kind of laying out the loss function, and you're looking at this k nearest neighbors approach for looking at how do you get the sort of the circuit the pairwise relationships within the hypergraph. Yeah, so that's good. Yeah, thank you.
Welcome, sir.
Yeah. All right. Yeah, so I think Shreyas, Varshith is here. Hello, Varshith. Oh, Varshith. Okay, Varshith, you want to say something?
So, did you look at the changes I have made to our paper?
Yeah, I did. I did send you an email, right? I know I'm a little late on that, but
So I have made the changes as you have said. I have also added the reference which you wanted me to add. So I have also changed the reference formatting as you said.
okay.
So doing it in Word it is a little bit tough. So I did it in LaTeX and I have made sure everything is perfect as you said.
Yeah,
So
yeah.
I just want you to review it and just give me a feedback if there anything else I need to add. Maybe I'll add it as fast as promptly as possible.
Okay, yeah, I think that's fine for now. Yeah, just send me a PDF of the LaTeX file, and I'll send it along. I mean, we'll probably have to do more changes later because it'll go through a peer review process. So, but that's like we don't know what
I
that
have
looks like.
already shared it to you in the email.
Okay, yeah.
So I have shared the Word document and the PDF document, both version 5.
Okay, version 5. Okay.
Mm.
Yeah, you can send that along. Send the PDF. You sent the Word version along, but I like the Word version so I can like put in notes because PDF doesn't handle comments well. But this, but I think we're at the point where you can just send me the PDF.
Yeah, okay, cool.
Thank you. All right, yeah, that's good. Yes, we're making progress on some papers and making progress on some projects. Did you have anything else to add, Varshith?
No, nothing right now.
Okay, yeah.
Thank you.
All right, Utkarsh, would you like to say something?
Yeah, am I audible, right?
Yeah.
I just had 2 doubts. One, is there any any limits on agentic coding for building the project in GSOC, or I would frame my timeline according to that, since of course I am able to produce more outcome with agentic tools, and I'm pretty confident that I produce good results with them. So is there some sort of limitation or anything in regards to that?
So yeah, we don't really have any guidelines yet. So it's like we're all kind of trying to figure out what those should be. Personally, for me, I would like for people not to do like the whole project in terms of agentic coding. And I mean, like you can create code, but you do have to spend time making sure that it works and then fitting it into the project. So I mean, you might be able to use agentic coding for kind of coming up with different formulations of things. But like what you've had there is what you have here is kind of the pieces that you want to build. And so it's a matter of building them out and making sure that they work well. Not only work when you execute them, but together in a package. So I mean, as part of this is kind of knowing what approaches might work. You might have like a couple toolboxes in mind that you want to work with, and you want to approach the problem with some technique. So you might, be able to mock up something where you ask it to do certain things and you get some code, and then you have to spend time reviewing the code, going over it, make sure it's robust code and everything else. So we don't really have any. I think maybe we're gonna have to develop some guidelines this year for this, because, and I don't know how successful people are going to be with this. So yeah, you're welcome to use agentic coding, but like we have to really be careful, not just about like whether it works and whether it's good code and robust code, but also like I said about scope creep, which is that you don't want to just start the ball rolling and you start exploring new avenues, and all of a sudden you're off your schedule. Because remember, I like what I said about the proposals. We need to stick to a very tight schedule, because you have 10 weeks to do things, and if you kind of go off track for a couple weeks, that's going to be bad, because you won't be able to get to your endpoint. Now, there's certain things you may want to follow up on, but those are things that you really have to kind of judge. Is this worth my time? Can I do this quickly? And the same applies to trial and error. So you don't want to like try 50 things, even if it's not agentic coding. You don't want to throw a bunch of things out there and say, I'm going to try 50 things and I'm going to like find the best way to do things. You really, when you're proposing work on a timeline, on a schedule, you want to maybe limit yourself to 2 or 3 candidates that you're going to try to implement during the coding period. And that may be, like as simple as getting a program running, or it could be as something like, working out like a prototype for each method and evaluating their performance. And I've had people do this in the past where it wasn't quite clear what technique they wanted to use, but they had candidates in mind. And that helps you sometimes, because sometimes the candidate that you had in mind doesn't always get you to where you want to be. And I don't think it's so much a problem here, because we have, we've already kind of worked out some of this stuff. But yeah, I would say, we're just going to have to keep an eye on this. I'm going to allow agentic coding, but we're going to have to be very careful. And, if you're working on a project this summer, you need to be like proactive with me about that. Be transparent about, okay, I'm using agentic coding for this. Let's make sure that we don't get off track. Let's make sure that it's of good quality, et cetera, et cetera.
Yeah, that makes perfect sense.
Yeah.
I personally use it, like I write down the abstract functions in Python, since I'm proficient with Python only. And like I do the math, I do the like abstract functions, and then I pass it down with a like context to my agent and have it implement it for me. I write down the long parts, then I like review it, I check it, I find out bugs, and I like write a report and like some relation or like fixes that it needs.
Yeah. Yeah. So yeah, we have an onboarding guide for DevoWorm, which we usually use in the community period to get people oriented. And I have to update that this year for a number of reasons, because there are a lot of things I think I need to add on that. But yeah, I think that's good. A good way to do this. Okay. I think Shreyas has not.
There's actually one more doubt.
What was that?
Like, can I have my proposal reviewed by you in my like? I wanted to like see whether my project timeline is like too much, or should I like scope it down more?
Yeah,
I had some doubts regarding that.
Why don't you just send me your project timeline and I'll take a look.
Can I mail it to you? Like, can you please share your mail?
You can, yeah, email. Yeah, email.
All right.
Yeah, yeah. Okay, yeah, and yeah. So anyone, if you have questions about your timeline, it's the best way to propose a timeline is just to have a small chunk. Like, think about what you're going to do from week to week. Think about progression. Like, if I make progress in this week, what comes next week? What comes next week? What comes next week? It's kind of like an agile stand-up where you're always thinking ahead 1 week, and you're thinking about the big picture, but you're thinking about the incremental progress. And then if something goes wrong, how do I correct for that? Do I do something if I get stuck on something, can I do something else while I'm waiting to figure out what I'm doing, or is everything going to be dependent on one thing failing or succeeding, or maybe that's a bad way to go. You have to think about all those kinds of things. So.
It's right.
Oh yeah. Okay, Shreyas, I don't think you've said anything in this meeting. I know you probably want to give an update.
Yes, hi. Am I audible?
Yes.
Hi, sir. So okay, I don't have pretty much. I just worked on the proposal. So I did send the proposal over Slack, the PDF. I've almost completed it. I've sent it for review. And yeah, I just mostly worked on the proposal, and I got like flow diagrams, all the codes in place in the GitHub. And yeah, mostly that. Nothing new as such. I just streamlined everything, put everything in the top, got things right mathematically, and put all the proofs of what I'm doing, why I'm doing, and how everything falls in place. So yeah, that's it. My proposal is in your Slack DM. So yeah, I guess I'll be getting to have a review from you. For the
Okay. Yeah, I'll take a look. At it.
Yeah, sure. But just that. I mean, nothing much. The proposal is quite exhaustive. It includes all my work, like whatever I've done and whatever worked out. So yeah, and it also has like timeline and what are my goals, what I am to do, what are the methods. Yeah, it has a, so mostly I just worked upon that for the whole week. And I devised the proposal. Just that. Yeah.
Okay. Sounds good. Thank you. All right, I think that's it for today. Thanks to everyone who was presenting and giving ideas and everything. So yeah, our proposals are due on the 31st. So we have next week's meeting, and then it's like the day after that. Make sure you know that I think the submission time is 1600 UTC. So that's important to get that time because it's not anywhere in the world at the end of the day. Like some conferences, you have to be very specific as to what time it's submitted. Susan, you had unmuted.
No, I'm just going to say bye, and I'll try and work on some of the code that I was given and see if it really does work.
Okay, yeah. Okay, all right, thank you.
Thanks a lot. Bye-bye.
Okay.
Thank you, everyone.
All right.
Thank you.
See you next week. Thank you.
See you.